In the final section of this chapter, we're going to talk about stem cells. So you may have heard about stem cells in the news, oftentimes when they talk about embryonic stem cells. So we're going to talk a little bit about what that means, why stem cells are so important, why they're such a big research topic, and kind of some of the things that they're actually looking to do with stem cells, because it's actually really cool what they're doing with them. But first up, specialized cells perform specific functions. Okay, so you're not going to have a skin cell producing the same kind of effect that a muscle cell will do. Okay, because the skin cells are specialized for kind of coating everything else, protecting everything else from the environment, whereas muscle cells are developed specifically to help with motion. Okay, so you've got all these different specialized cells that have their own specific functions, they form tissues, all this kind of stuff all together, but all of those specialized cells originally came from one cell, the fertilized egg cell, okay, the zygote. So cells develop into their mature forms that we know of in their tissues, okay, through the process called cell differentiation. Okay, so they become different types of cells through this process. So cells differ because different combinations of genetic information are expressed. So this was another thing that really confused scientists for a long time. It's that every cell has the exact same genetic information, but you have cells that are very different. So it means that they're not using that genetic information in the same way. So depending on how the cells differentiate, different pieces of the DNA will be activated and then expressed, and that will create the different attributes of those specific types of cells. Okay, so that's kind of tricky, and there's been a lot of research that's gone into figuring out exactly how the different regions are activated or deactivated. And sometimes those specific regions that are not useful for, say, a lung cell are permanently deactivated. They're not necessarily cut out, but they are put in such a way that they're not really going to be used ever. So a cell's location in an embryo as the cells are dividing actually helps to determine how that cell will differentiate. So we're going to talk about a couple examples here. So first up, if the cells are on the outside of the embryo, they will actually differentiate into skin cells. So that makes sense, okay, if you think about it. You know, what's on the outside of your body? Your skin, okay? And then you've got the ones that are found in the middle, okay, so kind of inside of the outer layer. Those are going to form into your bones, okay? All right, so if you think about what's in the middle of your body, it's your skeleton. Okay, so it makes sense, again, that those would differentiate into bone cells. And then you also have other cells that are kind of in between the outer and the middle, and those will form your intestines and your various other organs. Okay, so a lot of that kind of makes sense when you think about it. It's like what's on the outside of your body, you've got your skin, what's on the most inner part of your body, you've got your bones. Okay. So that gets us into what are called stem cells. So stem cells are really unique, okay? They have the potential to be any type of cell. Okay, so if you've seen the movie Zootopia, they say Zootopia is the place where you can be anything you want to be. That's the stem cells motto, okay? So stem cells can either divide, when they divide, they can either divide into two new stem cells, or they can produce one new stem cell and one specialized cell. Okay, so that specialized cell has gone through differentiation to become a specific type of cell rather than just a stem cell. So it's actually taken out that potential for becoming any other type of cell. Now it has its job and knows what it's supposed to do, and it's going to go do that. So stem cells have the ability to divide and renew themselves indefinitely, remain undifferentiated in form, Okay, so that's the 2 new stem cells, and then also develop into a variety of specialized cell types. So really any type of cell type you think of, if you trace all the way back through that cell's lineage, it came from a stem cell at some point. Okay. So stem cells are kind of a blank slate when they are in that stem cell form. And then this, through the different differentiational processes, it can actually be programmed into a specific type of cell. Okay, so they are blank slates. They can really do a lot of different things. The body generally will try to use them for healing different areas, especially if it's kind of a major injury. It'll differentiate a bunch of stem cells into that area to help replace the specialized cells that were damaged from the injury. So there are 3 different types of stem cells, and there are a couple very slight definitions between them, but makes sense in a little bit. So first up, you have totipotent. So totipotent cells can grow into any other cell type. Okay. Now what doesn't make a whole lot of sense at this point is that you also have pluripotent, which can grow into any other cell type except a totipotent cell. Okay. Now the difference between them is that totipotent is the fertilized embryo. Okay, so it's right at the beginning of a new organism being created. Pluripotent is after that those totipotent stem cells divide, they are going to produce pluripotent stem cells, which then pushes them along the path towards differentiation. And then you also have multipotent. So multipotent will actually grow into cells of closely related cell types. So say you have a multipotent blood stem cell, it could then differentiate into a white blood cell, platelet, red blood cell, various things like that. So break it all down here. We have totipotent, which is your fertilized egg. Again, it's pretty much just one cell, but it gives rise to all other types of cells, and it is a new organism. Okay, so when we talk about research, we're generally not talking about a totipotent because it's just a single cell. So it's difficult to work with just one cell. When we talk about doing research with stem cells, generally we're talking about pluripotent. Okay, in this case, it's showing embryonic stem cells. Okay, so this is kind of a very simplistic version of an organism developing. Okay, so it's an embryo that has divided several times, and now we have a mass of cells. So you can see the outer layer of cells here, okay, and then you have your stem cells in the middle. So those are the embryonic stem cells. They are pluripotent, which means that they can become almost any other cell type except for the totipotent. So this is basically once they reach pluripotent, they are going to continue on to making a full organism. Okay, they can't go backwards to just a fertilized egg cell. Okay, so totipotent leads to pluripotent. Pluripotent will then start to produce specialized cells, and then you also get your multipotent, which are adult stem cells. So these are closely related cells that can kind of change into each other as needed. Okay, so again, generally when we've talked about stem cell research in the news, generally they are referring to embryonic stem cells simply because they're such a good research tool because they have so much potential. Okay, because they can change into almost any other cell type. Generally, people don't talk as much about adult stem cells simply because they're already partially differentiated. So there's not as much freedom in what the stem cells can do. Okay, they are still useful, and there has been quite a big push towards working with adult stem cells instead of with embryonic stem cells. Part of that push is trying to basically go backwards in time, take multipotent stem cells, and then convert them back into pluripotent. And then basically you've taken adult stem cells and essentially created embryonic stem cells without having to fertilize an egg and then harvest the stem cells from the fertilized embryo, which, you know, gets into all kinds of ethical questions about when does life start and all of that, which is one of the reasons why embryonic stem cell research has been kind of frowned upon for several years. So there is that big push towards researching adult stem cells instead of the embryonic for that reason. Okay, so you can find stem cells in both adults and embryos. Okay, adult stem cells can be hard to isolate and grow. Generally, the adults don't have more than a specific number of stem cells. So if you think about the number of cells in an adult body, and you're looking for a very small portion of them are the stem cells. So that can be hard to isolate. And then they're already kind of set in something of a pattern. So they can be relatively difficult to grow. You also have to figure out different conditions that help the different types of multipotent stem cells grow and all of that. Use of adult stem cells to heal or create new cells in a patient may actually prevent what's known as transplant rejection. So I don't know if you'll know about the whole organ transplant system right now, but basically whenever somebody needs an organ transplant, usually it's kind of a last-ditch resort because the body, once that organ is transplanted in, the body will recognize that organ as foreign, okay, because it doesn't have the rest of the body's DNA and it has different receptors on the outside of the organ. So the body will think that, hey, this is a mass of foreign cells. I don't know what it's doing, so I should kill it. Well, when that organ is, say, a kidney or heart or anything like that, that can be, you know, pretty bad for the person. And there's really no way of telling your immune system, hey, this mass of cells is good. This mass of cells is okay. It's actually keeping me alive. Don't kill it. Instead, in order to get around that issue, people who receive a transplant have to be on what are known as immunosuppressancy drugs for the rest of their lives. And that basically knocks out a majority of that person's immune system, which is kind of not the greatest solution. It keeps them alive because the body will not have enough strength to kill off the new organ, but at the same time, that leaves the person really susceptible to any other kind of disease. So where you might get a cold and you're over it in a week, someone on immunosuppressancy drugs could get a cold and it might kill them. Okay? And we don't normally think of colds as being that dangerous. But to somebody who doesn't really have an immune system, it can be incredibly dangerous. So using adult stem cells to heal or even create new organs, okay, would get around that whole issue, because then you're using the patient's own cells to create a new organ. So you're literally growing a new organ from that person's cells, okay? And that way, once it gets implanted into the patient, their body recognizes it as self because it was grown using their own cells. It has their own DNA in it. And so then you can basically replace the damaged organ without having to get an organ from someone else, or in certain cases wait for somebody to die and then have them be not only an organ donor but also a match, okay? Because if you think about it, heart transplants come from people who died recently, actually, too. So this would get around all of that. It gets around the waiting list, because then you would only have to wait for the organ to grow instead of for somebody to give up an organ, somebody who matches you to give up an organ, because they do try to match DNA enough that the organ won't be completely rejected right off the bat. So there's a lot of stuff that happens with all of that. But we're going to stop this video here and then pick up in the next one.